modules community. I think the previous seminar that we had a month ago was really a great success. There are, very interesting talks, and it was really exciting. So, with this, I'm happy to chair the next session. I'm just sharing here some house rules before we get started. So please don't take any screenshots. Don't publish or post about any of the speaker's work, particularly if it's unpublished. And then when we get to the end of the talks, you can either raise your hand or use the Q&A function in order to ask questions. So with that said and done, I will hand over now to Dr. Dezső Módos. He completed his Doctor of Medicine and also his PhD in Hungary, working on network biology. Then he held several research positions in Cambridge and also in Norwich. And he's recently started a fellowship at the Imperial College London, working on network biology approaches to inflammatory bowel disease. So, Dezső, the floor is yours.
And hopefully you can see my screen, and hopefully you can hear me as well. Just double checking that everything is technically all right. All right.
Yeah,
fantastic. So thank you very much, Julian, for the nice introduction, and I am really happy to be here. So I want to talk about a bit of how you can use various network biological approaches to discover where the pathomechanisms and maybe some kind of therapeutic avenues to inflammatory bowel disease, or IBD. So the first question is, what is IBD? IBD is such a disease which has coming with tiredness, pain, digestion problems, and usually blood in the stool. These are the most common symptoms of IBD. It has a pathogenesis which is not exactly understood. We know that it has an immune component, it has microbial component, and the diet has a very big effect of it. And definitely it has a genetic component as well, affecting, for example, autophagy genes as well. Mostly of these genetic changes are in the non-coding region of the genome. So that's what I am brought to you to a bit more understanding of this part of IBD pathogenesis. It comes in two major flavors, Crohn's disease and ulcerative colitis. The difference between them that see the effects in any part of the intestine. Meanwhile, ulcerative colitis mostly affects only the larger bowel. CD is transmural. Meanwhile, ulcerative colitis only affects on the surface of the epithelial layer of the gut. It has effect of lots of people, more than 9 million people worldwide, and it's tending upwards towards the trajectory of the effect because mostly of the dietary changes in the developing countries, as like in China, for example. This is how it looks like more or less in an endoscopic big picture. This is the healthy bowel. This is the very inflamed red all over the blood and pus coming out of these ulcers. So that's not a nice look, and it can actually end up, for example, in colorectal cancers or very dangerous fistulas, which can kill even the patients. Why we need to work on it? Well, it has we cannot differentiate that what is the best therapeutic markers of this disease. We know that there are a lot of treatments. There is a big therapeutic arsenal, but we are giving it to the patients. They are getting a frequent relapse, and we know that there are a lot of single nucleotide polymorphisms which are affecting IBD. The more than 240, which are coming up, but we don't know that exactly how these SNPs have an effect in IBD, and are there any of them which can actually tell us that which therapy would be the best for the patients. So we have a very complex disease. It means that we will need some kind of complex methodology. The methodology which I am usually using to decipher the IBD pathogenesis is called network propagation. This is a very famous review from Cohen et al., or from which was in Nature Review Genetics, and here are that how you can use network propagation in the most simplest way, which I also used in a few of studies. I will show that one first. Is just we using the first neighbors. We have some kind of genetic exchange, a single nucleotide polymorphism in a gene, and then just check in a network their direct neighbors. Or we can walk around with a random walk with restart algorithm, and then we just with like a random walker, we walk in the edge of the graph at some directions, and then as the time is passing, we will basically get the weight of the graph telling us that which ones are more higher chance to get at the network. We will have t equals infinite, then we will have an equilibrium in the whole network at the end. And or we can use a heat propagation algorithm as well. Well, when we basically just have some source nodes, and then the heat is just stepping away from those source nodes and show us that which part of the network will be affected. And basically, if we'll use the random walk with restart and the heat propagation algorithm with a little cheat code that the heat propagation has a little dissipation component, then the two methods is basically equivalent. So I use basically a heat propagation kernels in my works most of the time. Okay, what kind of networks we can use for something like this? The most commonly used networks in my lab and in my works is usually protein-protein interaction networks. Here, the nodes are proteins, the interactions are physical interactions. We have either manually curated so that there were lots of small-scale experiments, and then they will some very clever PhD students read them, and then they put them into various databases. Or some large-scale experiments measure the physical interactions between the proteins, but we know that there is some kind of physical interaction, at least in one particular cell type. These networks can be directed or undirected, depending on that how much information we have, or can we predict, for example, the directions of these interactions. And usually what I am using is these networks are not weighted, so that we don't know that whether the interactions have strength or not. And we usually the positive and the negative interaction is, I am not often I just consider that they have an effect. In most of the cases, it would be a slightly more complex modeling. The network which I am using is the OmniPath, which has a relatively high coverage for the human protein-protein interaction network. That's the pros. The cons is that also it has good enough physical interaction, so they have high. I'm sure about the interactions, but it's not it's a bit uneven. Because if you imagine those proteins which we have researched a lot, such as cancer-related proteins, they will be highly overrepresented in such a network which contains a lot of curated information. The second type of networks which I am using is usually these transcription factor target gene network regulatory networks. Here the nodes are regulators, and such as transcription factors, in some cases microRNAs, but mostly here in the transcription factors. And we have their target genes, whether they are the transcription factor target genes or the microRNA target genes. The directions here are directed, and they are regulatory interactions. So for the regulators, we go to the regulated target genes, all the cases. The network is a directed bipartite graph in this case, with a little catch that the transcription factors can be as well their targets as well at the end. So when we will move the bipartite parts together of these networks often. One of the network which I am using in the Dorothea network, which you will see in the result slides as well, and my parent lab, Tamás Kocsmáros's lab is working very closely with Julio Sáez Rodriguez's lab, and that's where these very cool tools are, and which we have developed these together, particularly in the OmniPath. I was also participating in the development and mostly the testing process of it. But they are very great tools for using network biology. I brought you two questions which I am trying to answer. were using these kind of methodologies. How can we cluster ulcerative colitis patients based on protein-protein interaction networks? Can we get some kind of information back that the pathogenesis of ulcerative colitis is different when we are looking at network biology? And the second one is, can we find some cell specificity when we're looking at networks in Crohn's disease? That will be the second part of the story. So, what we will start with, we have some single nucleotide polymorphisms from GWAS studies. So there was a lot of genome-wide associative studies where they measured that a single nucleotide polymorphism actually tells the differential expression in this disease, in this case ulcerative colitis, in a certain gene. They collect all of these. Then we have a few immunochips from patients, in this case for 378 patients in this network cohort, and we are checking them that whether they have these particular single nucleotide polymorphisms. As I said, most of these single nucleotide polymorphisms in the regulatory regions. When we have a single nucleotide polymorphisms in a regulatory region, then we have to figure out that whether that actually has an effect on some kind of gene. How we can do that? We check that whether that is in an enhancer, and we can find a transcription factor binding site which alters in the presence of the SNPs, which will create a new transcription factor binding site or makes it inaccessible by the transcription factor based on the sequence. The same with the promoter, so the gene before the gene we can check as well the promoter sequence for this. Or, which we tried as well, in the coding regions or in the first intron, check that whether a microRNA target site will change when we have a single nucleotide polymorphisms. And with that, a new microRNA will be able to bind there, or this old microRNA will not be able to bind there anymore because of the SNP. So this one we will use to identify the gene which will be affected by these transcription factors or affected by these mRNAs. When we have these SNP-affected genes, then we can map that to the protein-protein interaction network for each of our patients separately. And then we can use a very simple network propagation algorithm, such as the first neighbors. We will just check that what are the interactor partners of these SNP-affected genes, and from that we will be very, very happy. We will have a nice network which will have all the interactor partners. When we have this, then in the real life, so we can check that back to the patient cohort and then try to figure out that have we find something interesting, have we find some relevance with these networks compared to the patients which we have. When we will look at it into the real world and not just on a figure, on a simplified figure, this is how it looks like. Here we have those transcription factors which have altered their single nucleotide polymorphisms, their binding sites. Here we have the microRNAs which have a SNP altered their target sites. Here are those genes which these microRNAs and transcription factors are affecting. And then downstream from them is we have their interactor partners. You can see these nice colors. These are coming from a very simple check of what are the main first neighbors of certain genes. Basically, that's what we have used in this case, if I remember correctly. And then when we have something like this, then we have to figure out that what the heck they mean. And for that, we were just using the good old-fashioned gene ontology biological processes, and then we can give them specific work areas that what they are doing. So like this one is doing calcium homeostasis. This part is involved in cell motility and adhesion because of the VEGF SNP-affected genes. These are involved in antigen presentation because these genes are HLA genes, so human leukocyte antigens. Here we have some methylation and other stuff, and here we have mostly stress regulations, including autophagy and the NF-kappa-based signaling as well. Okay, great. We have a beautiful network, but how we can get back to that to the patients? Well, if we take each of these nodes and then write up that whether it's involved in a particular patient, then we can get a nice heat map. Here we have each of those proteins which we have seen before. And here we have each of those patients, and here we have those big hubs which were affected, such as the HLA, the antigen processing, VEGF, which I mentioned, SEP-BP and PTPN, which are involved in the cell migration and stress response, NF-kappa B, protein kinase C beta, which was on the left side of this network. Then we can see that how when we will cluster the patients, that what part of the effect they will be actually really affected. And we can see that, for example, these patients, these reds ones are involved in stress response, FC receptor, these greens calcium-mediated pathways, cell migrations, germs, and these are the rest of it, other kind of pathways. And when we have this, we can actually visualize them into the network in network ways. So here we have the first cluster where we have the protein kinase C beta, SEP-BP, PTPN1, HLA, either positive or negative networks. And then we can see that with the protein kinase C beta positive, but PTPN, SEP-BP and PTPN negative. So we don't see these nodes here. Then protein kinase C beta negatives, but SEP-BP and PTPN1 positive. So this one is actually the nice and yellow. And when we have both negative and both are blue. Okay, these are you can say that Dezső, you have showed us very nice network pictures, but what do they mean? Because that was just not much clinical relevance yet. Well, we need to validate this kind of efforts. That do they show something? How we have done that? We have taken these SNP-affected genes, and we have used a completely different cohorts where we have treatment naive UC samples, and then we taken there that whether the here the transcriptomic analysis comparing to the non-UC patients, do they have actually the differential expression shown in certain SNP-affected genes? Do they have when they cluster these patients, based on their SNP-affected genes, and their first neighbors, so that based on this yellow network, are they get a similar? Can we recognize the clustering which we have got as well? Well, so that was the two-way how we tried to validate these things, and we have also done the pathway level similarity checks, but I don't want to show that one because I think these two are much more clear validations. And what we've seen that, okay, in this completely different cohort, juvenile UC, we have most of these genes which were SNP affected, they are actually differentially expressed. Anything which is yellow is differentially expressed based on fold change in compared to the control, and the numbers are that in how many of these control cohort, or they are actually differentially expressed. So yay, we have at least a lot of them which are we can see that including the protein kinase C beta, which was one of our main SNP affected gene, NF-kappa B, which we have expected, SEP-BP as well, VEGF, PTPN, great, PTPN1, great. And we checked as well that how we will get that when we have these hubs and their first neighbors here, and are they first neighbors differentially expressed? It's and how much of them. Here the picture is not as clear. We have got one cluster which is very nice because it's almost all of their in these first neighbors and their interactor partners are differentially expressed, but we also get others where, well, not so much, so that they are not much specificity we have. Sadly, we didn't get back the protein kinase C beta, and which was just that one was very, very yellow, but we have got back the NF-kappa B one, which is one of the main transcription factors which actually involved in IBD, so that their interactor partners in a protein-protein interaction network, not in a transcription factor target gene network, is as well differentially expressed, at least in a set of patients. So, what we have learned from this, we have basically identified that the regulatory SNPs, when we will follow them to one step even, when in UC they have identified pathogenetic processes, and they are hubs which are driving the network clusters with these first neighbor networks. So if we have a hub, we're hitting a hub with a SNP, then they will have a very strong effect in there. We see that the transcriptomic and the network clusters were relatively similar, so that we can say it's great. And one of the important things is that UC is still very heterogenic. And when we are trying to figure out that which is the best treatment, we have to be taking into account that what is the genetic background of our patients, because otherwise we will just cannot do a one size fits all method. The problem with this is that it's only a PPI network, and we have a very strong hub effect, and we don't have a cell-specific outcome. So what we done next in Crohn's disease? Here we have again the same story so far. We will have the SNP-affected genes, and then we will have the signaling network. But here we will do a different approach. We will do actually network propagation on here on this particular signaling network. And then when we reach here the transcription factors, we will move them to a regulatory network as well. And in those regulatory networks, that what will be the predicted transcriptionally affected genes, and use them to actually do the clustering and try to figure out that what we will get. So let's see what we will see in this case. Here we see the signaling network back of these Crohn's disease patients. They converge on cell cycle and apoptosis. Here I have used just a Green-Newman clustering of the network, and then we can see the different protein-protein interaction modules. We can see that what each of them are involved in apoptosis, immune response, cell cycle, regulation of cell migration, and we have even found some autophagy-related module as well. And then we can go downstream from here, from these transcription factors, and see their interactor partners as well. Here we have, these are the transcription factors, and here we have their target genes. So we have now a regulatory network. Before we were a protein-protein interaction network, and now we are in the regulatory network in this case. We can see that here we have the MAPK signaling in the ETS1 and ETS2, which were described in a recent paper that they are important potential drug targets even in CD. The STAT3, BRF, SMAD3, P53, we have got some random reasons as well. And then what we've done is use these networks and try to do a clustering, and basically stratify the patients. We have identified 12 clusters, and we have found that which transcription factor will be common in one of these clusters. Here you can see based on the Jacquard similarity matrix, the PCs of these clusters. And when you have this, then you can write it down that which is each of the clusters have, which each of them have their regulatory regions, and which transcription factors are actually active in them. When you have this and visualize them in a network, can check that whether which of these network will be actually overlapping with a cell-specifically differentially expressed genes. And that's what we did. We've taken another data set from the Congetol data set and see that whether the specific cell in a specific cell type are the differentially expressed genes actually overlapping with the genes in a particular cluster, which are more common than 0%. And we get them back. Yes, there are cell-specific differences. You can see, for example, that the ETS1, ETS2 cluster is more of in the T cells, DC1, DC2 cells, and not in a B cell or mast cell cases. And we also seen that with the colon, where we have got actually a slightly different picture. We have got these goblet cells only in the ETS1, ETS2, and STAT3 network. Meanwhile, when we have only low number of targets in the transcription factors from one clusters we get back. The only one which was missing is the p53, which was not surprising, to be honest, in the long run. So, in summary, these SNPs and SNP co-occurrence is more relevant one marker than one SNP. Seems that that's so. And we also need to go downstream to actually increase the opportunity and stratify our patients better than otherwise. Again, we need to differentiate between the different type of patients, and we can connect the IBD-associated pathways with networks, and if we will take the regulatory SNPs. And if you want a bit more, we can read the paper which is now in medRxiv, where we actually went for not just the cell type specificity, but also checked for drug response in ulcerative colitis, and maybe these kind of mechanisms can uncover some resistant pathways, because it's one of the biggest issue in IBD pathogenesis. So where next? We want to do that and include for epigenetics and clinical metadata for omics, and we also want to do single-cell RNA-seq for understand then the network propagation for those data sets, and include that for therapeutic response as well, which is one of the most important thing that we want to use this kind of studies in multi-omics and regulatory networks. So this is the team. I was part of the Tamás Kocsmáros's lab. Where is Tamás? Here is Tamás. In a long time ago. Well, not so long time ago, but and a lot of people here who are involved in this project, in the iSNP project, and now I am here thanks to the Imperial College Research Fellowship for having me here in the Imperial College, and now I can talk to you, and maybe I will have my own lab soon. Well, I have my own lab, so I just I can include lots of things. And for a bit in news about, there will be a cool conference this year in Visegrad. If you want to come over, we're into Hungary, and this beautiful place, and have some fun and lots of interesting talks about IBD and signaling networks. Come here and enjoy. And that's all. I am ready for your questions, and I'm sorry that probably I was a bit over the time.
Something quite different. So Dr. Joanna Congella is currently a postdoc in Glasgow with Helen Walden. She's been working on protein ubiquitination for a number of years now, and is now working specifically on Parkin. So take it away. I think, Dezső, you also need to stop sharing.